Thank you very much for joining us today for this webinar. The title of the webinar is Ligand-based lead discovery using atomic property fields. So if you have any questions during the webinar, please feel free to use the chat option where you can type in a question, and I'll try to answer it during the webinar, or at the end we'll have time for questions. If you want to be on our mailing list, please click on our news link here on our webpage, and you can also follow us on Twitter and Facebook. So today we're discussing atomic property fields, which we'll refer to as APF. So APF is a 3D Pharmacophore which replaces discrete points by continuous chemical property distributions. So APF replaces representations of chemical moieties with fixed Pharmacophore types by vectors of atomic properties that can be compared in a more flexible quantitative manner. This method is fully described was developed by Max Totroff and is described in this paper here in 2008. How can you use APF? So it has a number of different utilities. So the APF pharmacophoric potential is implemented on a continuously distributed grid and can be used for superimposing two ligands or multiple ligands using multiple compound alignment. You can perform ligand-based virtual screening, whereby you would have a set of ligands that you know bind or are good binders, and you can use those fields of those ligands that are superimposed and then screen millions of compounds onto those fields and get a score, and you can rank how well other chemicals in your database fit into that field. So you can also incorporate, so there's no protein structure involved in that method, but you can incorporate a protein boundary around the ligand, which would give some penalty for the ligand going outside of that boundary. You can also use APF for 3D QSAR. So if you have your ligands and also maybe some binding data, you can build a model that could link the binding data to the atomic property field properties. And kind of separate from this, if you have two proteins that bind similar compounds but don't have any sequence similarity, you can use atomic property fields for superimposing protein structures. And one other additional utility of APF, which I didn't mention here, is that if you have a hit list of compounds from virtual screening, you can cluster by atomic property fields as well, which we'll show. Okay. So these are the potential components that we include in APF. So we include hydrogen bond donors, hydrogen bond acceptors, regions where sp2 hybridization, lipophilic regions, a field for size, and we also have charge, positive, negative, and also electropositive and electronegative regions. It's just an example with one compound that has many of these properties in it. Okay, so in this webinar we're going to show this in ICM. This can be done with ICM Chemist Pro or ICM Pro plus Chemistry. I will show how to superimpose chemicals using APF. We will show how you can show these fields for the superposition. We will show how you can get a score for two superimposed chemicals, and how to perform virtual ligand screening using the fields, and also how to use the fields in the ligand editor, where you can edit the ligand in 3D, dock in the fields, and minimize and make changes to the compound. And also show how you can superimpose binding sites as well. Okay, so in this example, we have two chemicals that have been read into ICM. So you could read them in by going File, Open, and an SDF file, or you could sketch them, for example, using the sketching tools in the molecule editor here. Okay. So once I read them in, it's important to convert the ligands to an ICM object. This will give the atoms the correct types, and you can fully superimpose. So these chemicals, atomic property field superposition is ideal if you have chemicals that have similar properties but different substructure. If they had the same substructure, you could quite easily use the chemistry superposition by substructure options here. But in this example, there's only one conserved region, but the rest have similar Pharmacophore properties. So we're going to superimpose these by atomic property fields. So we need to go to the chemistry menu, we go to APF tools, and pairwise, so superposition, and then pairwise APF alignment. And the dialog box uses a new graphical selection that we implemented about a year ago, which are these orange crosses. So it's a way of having two concurrent selections in ICM. So I'll just show you how to make an orange selection. You double-click on a ligand, on an object, and you can see it's the standard green crosses that we usually use for representing a selection. And that standard green crosses goes into the variable called AS_graph. So we could say display CPK AS_graph, like that. But if the new orange selections give you a bit more flexibility, and enable you to make more than one selection in one go. So we will convert this green selection to an orange selection by clicking on this button here. And you can see that the green crosses are now orange. And the variable for this selection is AS2_graph. So if you want to display in CPK, we'd say AS2_graph, like that. So we have one selection, and the other selection is a standard graphical selection. So we just double click on the ligand here, and we can choose to superimpose both objects, allow both to be fully rigid, or allow one of them to be static, or we can have use flexible alignment. So I'm just going to use the default option. Thoroughness will increase the length of the simulation, trying to fit them in, and you probably get a slightly better fit if you increase that. But for this example, we'll just go OK. It's superimposed those two ligands which have similar properties pretty well. So we can visualize the property fields that we just used to superimpose by going to the chemistry menu, APF tools, and there's an option called consensus Pharmacophore, basically. So here we need to make a selection of the ligands that we want to display the fields for. So we can do that by double-clicking here to select both of the ligands. And then you can set, if you had more than two, or if you had many compounds superimposed, you may want to take a threshold and say, I want to display the donor field for if 75% of the ligands contain a donor in this region. The same with the acceptor, charge, and hydrophobicity, so you can go OK. And then you have displayed the property fields here. So we have the hydrogen bond donor region superimposed well here. We have the hydrogen bond acceptor region here. And we also have these lipophilic blobs and another acceptor here. And then we have this the sort of aromatic conserved region here. Okay. So we've superimposed them. We may have like a database of 50 or so compounds, and you want to see which one superimposes best on your lead compound for further testing. So one way to differentiate which one fits well into the fields is to use a score. So we can score how well these two ligands are superimposed by going to the chemistry menu again, choosing APF tools and Pairwise APF Score. There's a similar process as to when we made the selections. So we need to convert one selection to orange selection, or the second clicking in there, and then we make a selection of the other ligand, and we go OK. And this produces in R_out the score for these two compounds. So the lower the score, the better the fit in the atomic property fields and the superposition. Okay. So the next example is we have these two ligands. Maybe we have more that we know bind very well to our receptor, and we want to screen a database of millions of compounds to these fields in order to find new compounds to test to see if we can improve the binding. So what we would do, if we had more, we would superimpose more on top of each other. If, for example, you had a crystal structure with the idealistic pose, you can use that as a template and then superimpose the others on top of that, and you have a nice grid for screening. So in this example, we are going to use a database, small database just for the example, and to find it. So the database, we just go File, Open. It's in SDF format. I just want to show you the database before we screen it. So this is a database taken from our MolSoft database, or you can download SDF files directly from the vendor, or there are other websites which have the SDF files for you to use. So yeah, we have, and just for this example, we have 15 compounds that we're going to screen to these fields. Here. So to do this, we go to the chemistry menu again, go to the option called APF fields, and then screen SD file. So it's asking us where the SD file is located. So you have to browse for that. And go open. So the path is here. It's asking us what ligands that we want to use in the template to screen against. And it's a selection, so we select these two. You can use the control key to select one, the other one, or we can just double click on objects because we need to use both of these objects. So as before, there's a thoroughness value, which is the length, which is going to represent the length of the simulation that you're running. If you're screening millions or hundreds of thousands of compounds, you may want to keep it at 1 just for the speed. But this is very quick actually. It's pretty less than 5 seconds per compound. But if you wanted it to be more thorough, you can increase this. Choose the sample rings during the simulation, sample cis and trans, sample tautomers, which will take a bit longer to run. So you can have 2 different scores. One, the APF score, and the other is how well it fits inside the actual density. You can have a cutoff, so it will only keep scores that are less than, that are less than -100. And there's also other ways to. You may have atoms where you're unsure of the correct position, and you can lower the occupancy for these to 0, and so it will be ignored or to a different weight. You can also weight by lipophilic regions and the polar weight as well directly. Okay, so once we're set up, we can go OK. So this is screening 15 compounds. You can see that there's a progress bar here. You can also view the progress by going Windows, Background Jobs, and you can see it's been running for 10 seconds. And so in 10 seconds, it's finished screening those 15 compounds. So it's significantly faster than structure-based screening, for example. So so now we have, when we pressed OK, we have the hit list, which is just given this name based on the ligand name. And we see that we've kept the same rows from the original database, but we have an additional row column called score. So if we wanted to sort by score, we can right-click and choose sort, and so it's ranked by, the table is ranked by score. If you want to view how well these compounds fit in the field, you can click here, and the top hit's very good. It's the yellow one. You can see it fits very nicely into the field. And as we go down the hit list, the scores are pretty good. Here, the scores get a little bit weaker because it doesn't quite, match the whole template. But significant regions do, so that it's still a reasonable score. And you can go through here. And the lowest one is this one, which doesn't contain this region here. Okay, so that's virtual screening by APF. If you want to run this on the command line, you can do that as well and split up the jobs onto separate nodes. And the macro we use is called, the program is called Chem Super. I can just show you that. So yeah, it's called Chem Super. So you would, on the command line, you would use this. You would have your template object. So you would save, for example, the 2 ligands that we use in our template. You would save it as an object file. So you'd select them, and then right-click and save as ICM object, and call it template, for example. And then you define where your SDF file is located. You would ideally use an index file if it's a very large database. You can index the file by going to Docking, Tools, Index, mol2 here. Then you have an output file name, and then you have other parameters that you can use. For example, sample the rings, rigid superposition, and such like. So that's on the command line. You can run it in batch. Okay. Right. So the next example is how to do this in the ligand editor. So okay. So in this example, I have 4 indomethacin cyclooxygenase inhibitors, and they're all very good binders, and they're all superimposed based on a crystal structure pose. And so they're called template 1, template 2, 3, and 4. I also have a ligand that I want to dock to these fields, and also edit and try and improve the fields based on using the atomic property fields. I also have a receptor which we could use to define a penalty around the ligand. So we wouldn't want to build out here because the receptor is there. So to do this, we use the ligand editor tab. First we need to set up the ligand, which is called ligand. So we use the drop-down button and identify the ligand. Just for clarity, I'll remove all the other templates. So this is the ligand we want to optimize and dock. And go OK. And then the next button along in the ligand editor is the receptor setup. So we press here. And usually, if you're doing structure-based optimization, you'd use the protein tab here, which sets up the receptor. We're going to use the APF tab. Okay. So you can see that it's asking us for the template molecules, which molecules are in our template. We can weight the parameters for lipophilicity, polarity, and other options. And we can also use excluded volumes. This is the way to incorporate a penalty for going outside of the. So we could use the receptor as an excluded volume here. So we'll for example, we'll do that. So we need to make the receptor an orange selection, and then we need to select the templates that we're going to use as the field. So we click here, here, and here. Okay, so now we have 4 regular selection green selections. So we can drop down and choose selection 4 molecules here. And if we use graphical selection, this is the receptor. And we'll make a box around the ligand. And it's the purple box, which you can drag here to make it larger or smaller. And then we go OK. And so this is building the APF fields inside the receptor. You just look at those maps here. So this is where the ligand is going to dock. Okay, so basically we're now editing. We can edit and dock this ligand in the atomic property fields. In the ligand editor, there's a way to display the atomic property fields. You can visualize the APF here. And we can also visualize the excluded volume that we use as well, which should give you a way to see where you've got cavities where you can build also. Okay, so the ligand is set up. We can minimize the ligand in the fields by clicking on the minimize ligand button here. You can see we have a score here. We could also dock this ligand, which is a longer simulation. And if you were here for the first webinar in August, we showed how you can edit a ligand using the ligand editor, and you click on here and you can make changes to the ligand in the atomic property fields. So you can add make single changes by clicking on a substituent and clicking where you want to change, then evaluating the score. You can delete atoms in the same way as before and evaluate the score. So the score's got worse when it's not fitting in there. You can screen multiple substituents in one go by selecting different ones and then just clicking on the point where you want to make the substituent change. You can edit in 2D as well if that's easier by clicking here and deleting atoms that you're not interested in. Okay. Or you can click on the Find Group, and that will screen a small database of substituents at a particular point in the atomic property fields. Right, so that's in the ligand editor. If you want to see more about the ligand editor, the webinar on the ligand editor is online in YouTube at the moment. So the next example is sort of kind of moving away from directly superimposing ligands. It's a way to superimpose proteins that are by sequence they're not similar at all, but they bind the same ligands with similar properties. So you can superimpose the binding pocket doing that. So there's an example here. Okay, so prepare this. So here we have two proteins. You can make it easier to see them by going color by objects, and this will give the two objects different colors. So there are two proteins from different protein families, but they both bind this this ligand, thiamine diphosphate. Two phosphate groups here. Okay, and so the idea is to superimpose these proteins. So if you try to do it by standard ways that use sequence similarity or 3D secondary structure similarity, the pockets won't superimpose at all. And we can see for example, we align the sequences of these. We extract the sequence of one and extract the sequence of the other, and we can align those. You can see from the alignment that these proteins are not similar. Okay, so the aim of this example is to superimpose two pockets by atomic property fields. And Max Totroff, who developed the APF method, has also published a paper on protein superposition by atomic property fields as well. And I can send you that that paper as well if you're interested in looking at it. So to superimpose these two proteins, we go to the Tools button and then Superimpose. Tools, Superimpose. There's an option called Sites by APF. Go OK. And so we choose which protein is going to be the static one, So we can, just for clarity, we'll also delete the other subunits, other molecules that we're not interested in. So all we really need is the receptor and the ligand. So the other molecules we can discard. Okay, so we make one of the ligands an orange selection as before, and we double click on the other ligand. We go to the Tools menu, Superimpose Sites by APF. You can see we have two selections. One is the static and the other is movable. So this is static and this is movable. Then we go OK. Okay. You see how well the two ligands are superimposed in the pocket, even though the pockets, because the pockets obviously have the same properties because they bind similar ligands in a similar pose, and so this is a way to do that. Okay. Using atomic property files. So the last example is clustering, chemical clustering. So last week we were showing virtual screening, and so I have an example with a hit list there. Okay, so this is an example we did last week, and everything is saved. So I can go to Open Project. And the project name that we called the docking project was Webinar. We go OK. So that reads in the receptor that we use. And then we can remake the hit list by going Docking, Make Hit List, and go OK. Okay, and for that example, we only screened 50 compounds, so we've got 50 rows in our table, but we could maybe envisage that there's going to be a big hit list that we want to select, maybe 200 compounds for experimental testing. So one way we could do that would be to sort by score, for example, and take the 200 best scores. But most likely in the 200 top scores, you have similar compounds, maybe with similar substructure, and also binding pose. So ideally, you'd want to select 200 diverse compounds, from the hit list, rather than repeatedly. If, for example, one doesn't work, you then waste money by buying compounds with similar properties. So you could also do a fingerprint clustering, but in 3D you could use atomic property fields to cluster, and then you can select a diverse subset. So here we have 50 compounds. We can use the option here, cluster by APF, which is inbuilt into the hit list. Or we can go to chemistry and choose the option chemical APF tools, APF clustering, and it asks for the chemical table. So any chemical table that has 3D ligands superimposed, it'll be able to cluster for you. If you have more than one, if you have a very large table, you may want to use all available CPUs here, and go OK. When we do that, we get a tree shown here, and we can change the distance between these by dragging on this bar here. Okay. And to select representative centers, we right-click here and choose the option Select Centers By, and you choose by, for example, minimum score. So for each one of these clusters, we will take the representative center, which will be the one that has the lowest score. You can choose how many you want to choose. So you may want to choose one from each cluster, which is the idea. Or if you have very large clusters, you may want to take more than one from each cluster, which is an option here. And there's other options for how to treat singletons in the tree as well. So you just go OK, and you can see in the tree has made a selection here. The blue ones are the centers. So we can right-click on any blue one and say copy selection to new table, and you can give the table a new name. Hits for testing, and these are a representative sample of compounds, diverse set for experimental testing. Okay, so that's really the atomic property fields and all the different options that are available. The option I didn't show was the way to build APF QSAR model, but it's relatively straightforward if you have the data. So for the APF 3D QSAR, you need to build a model based on a particular column. So you could use score, but you may have like experimental values here, and then it would build a model using partial least squares, and then you can use that model to further to build another score, maybe for testing based on the QSAR. Okay, so if you have any questions, please feel free to write them in chat, or if you want to speak, let me know and I can unmute your microphone. We plan to have some additional webinars in September, but we're looking for suggestions of topics that might be interesting in. So please email me some ideas, or you can use the chat also to let me know if there's a particular topic that you would like us to cover. So once again, if you want to give us a call or email, please do so. Thank you very much for your attention. We had a question about the papers for this method. The original paper is here. The one describing ligand binding site superposition is in BMC Bioinformatics. Yes, so it's top off atomic property fields. Or I can send you the papers as well.